good evening, everyone. I'm very happy to be part of this discussion. And as if, I've been told that there were series of discussions on the idea of evolution in the past, and I've been asked to discuss something about the Neo-Darwinism. And certainly, this topic, Neo-Darwinism, is a huge topic, but I hope I'll be able to touch upon it a bit, and then I'll try to discuss. Yeah. So I'm working in the butterfly. So some of the slides are slightly biased towards butterfly, but I've try to incline it with the line of discussion that we are supposed to do today. So yeah. So I'll begin and. Firstly, I'd like to briefly look into the nature and appreciate the diversity and the variations, and then maybe I'll look into the some idea of Darwin and the challenges that were there during those period of time, and then the discoveries, or let's say the understanding of genetic variations and the various mechanisms which can lead to the variations, and also some mechanism which shapes the phenotype of the organism in the populations. So since this discussion might include some of the technical terms, and if you find it difficult to get it, maybe you can just stop me and ask. I'll try to discuss that in more simpler form as much as I can. Yeah, I'll start. Yeah, so it's very obvious when we see around, right? When we step out of our home and when we see around us, there are different types of life surrounding us, right? So, like the life surrounding us is very diverse. Like within plants, if we see, then like within tall trees itself, we see like there are different types of trees. Insects, there are huge diversity, right? Fish, reptiles, and birds, and all those things. Like there is a huge diversity of life that surrounds us, right? That's only that is apparent to us. There are many things that's very tiny, which we can't see, or we can't see them through our naked eyes. But yet, the diversity of life in that microorganism level itself is very huge, right? So there is a huge diversity, and this diversity of life that surrounds us is really fascinating. Now, when scientists looked into this, and when naturalists, when they started to categorize them based on the similarities and the differences, they tend to categorize it like this. So this is basically a pie chart here. They have categorized different organisms based on the similarities and the differences that share among themselves. So this is one of the representation. So definitely, again, my point is to make it appreciate the fact that the diversity of life around us is really fascinating. Now, again, this is the one slide which I thought I'll put it to bring into the discussion one important aspect, that is, if you see these four different butterflies here, then these all of this like these four different individuals apparently may look like same. They may look like belonging to same species. So when I say species here, basically what it means is two individuals. If I put them for a breeding, then two individuals will be able to produce the offspring, and that offspring will live up to reproductive age and reproduce the viable offspring. So if they could do so, if two individuals can interbreed and produce the offspring, then I'll call that to be a species. So here, these four individuals here, they apparently may look like similar, but when we look into the details, so what we see is that these four different individuals belong to different species. So there is a diversity, and there is differences, and there are similarities as well. So the life that surrounds us, there's the differences and the similarity. That's one important point which I wanted to make it up to here. Now this is only up to like the species level. When we look into the within species, within population, in a sense, like let's say, so here it's a cartoon representation of the beetle. So here in this case, I'll try to. Yeah. So here in this case, this is the population of a beetle, and here you can see that there are two different color of the. There are two different types of beetles. One with they are same species, but there is a variation with respect to skin color or coat color. So one beetles, they have this brown color, and the other beetles have this green color. Now there's a variation with respect to color. So here the color of the body, I'll call it a phenotype. So phenotype, when I use the term phenotype, basically what it means is the character that we can observe. In a simplified form, the character that we can observe here, this the color of the coat of this beetle will be a phenotype. So what we can see is that within this beetle population, there is a variation with respect to color of the coat. Similarly, this is again representation of a mice. So here what we can see is that again here also there is a variation with respect to the color of the skin. So some of the mice, the color is black, and many and few of them have this tan color body. So there is a variations between species, definitely, but within species also, when we look into it, within population also, when we look into it, then we can see the variation with respect to the characteristics of our interest. For example, here we are looking to the characteristics of the skin color, right? So yeah, again, this photo, maybe you will recall it, this is the photo of a single species. So all of these individuals here, from this left to the right, when we check it, all this, they belong to the same species. They are mostly found in the same season of the year. For example, this dry season that's there, that's coming up in Kerala. During this time, we'll see this particular variation, this particular types of this seasonal form of this butterfly. So within this season itself, what we can see is that there's quite a lot of variation with respect to the wing pattern. So there is a variation between different species, but there is a variation within the same species as well, within a population as well. In the same season form itself, there is a variation in the phenotype. So what causes this variation? It is very apparent to ask, right? When we look into the nature, it's very obvious question to ask: what causes this variation in the traits? Now, at this moment, after posing this question, I wanted to go back to the idea of Darwin a bit, and then maybe I'll come back to this question to discuss the various causes that we know at this moment to understand, to explain the variations that we see. Now I'd like to go back to the idea of evolution that was mostly proposed by Charles Darwin and the contemporary biologist. Now, what Darwin discussed about this idea is basically the species. Now, when I say species, again, species are like individuals who can actually interbreed and produce the viable offspring. So they change over time. So when I say they change over time, what does it mean? I'll discuss in the next slide. And new species. When we look into the nature, what we see thousands and different types of species, right? Again, the question, very obvious question, is how this so huge diversity came into being. How it came. He tried to explain that all these lives that we see around us, they came up from the pre-existing species. And if we trace back the ancestry of all these species, then what we can see is that there was a time when they all originated from the common ancestry. So he came up with the idea of tree of life, right? So that was these are the basic ideas which Darwin came up with. Now, this species can change over time, or the compositions of the phenotype in a population, it changes over time. I'd like to explain this. I'll try to explain this this idea with respect to this very simple cartoon example. How is my is it audible now? It's clear now?
Now it is clear.
Okay, yeah, thank you. Yeah. Okay, yeah. So here, this bird you see, this is the predator, okay, and this mice, this is the mice, and you will see that there are two different types of mice here. They belong to the same species, but there is a different like few individuals they have tan color skin, and a few individuals they have a black color skin. Now, please focus on this picture for time being. So this predator, they feed on, they predate upon this mice. Now, the background that you see here, the background is let's assume it to be a rock. Now, the predator that is flying in the air, for this predator, it's very easy to spot this tan color mice compared to the black color mice, right? It's very obvious to us as well, right? So what will happen? So these predators, here I'm just represented, like I've just taken the example where only one predator is shown, but there could be many. They predate upon the this tan color mice because it's visible compared to the other individuals. Now, after it being predated upon by this bird, or let's say the predator, in the next generation, the ratio of individuals with the tan color versus the black color, if you see, then it's changing, right? Now, after few generations or immediately after next generation as well, what we can see is that this ratio is changing further. So here, this idea that I kept it here, the composition of the phenotypes in a population changes over time is basically, this is what I wanted to represent here. So here the ratio of tan versus black mice over generation changed. Okay, and another interesting thing that, yeah, which in a simplistic term we can also use this example to understand the idea of natural selection. So here, when I'm discussing the natural selection, I will also like you all to appreciate the importance of variation for a natural selection to act. So here, initially there was a variation with respect to color of the skin. Now, since there was a variation, few individuals had higher chance of surviving and reproducing. So here in this case, this tan color mice, the black color mice, they have a higher chance of surviving and reproducing because they are camouflaging with respect to the background. Now, because of this advantage, they could interbreed. They could reach the adult stage and they could reproduce. When they're reproducing, they are producing the offspring, and many of which they have the black color skin, like the fur. Now, this could happen because the characteristics of our interest here, this color of the mice, was transmitted. The information that is required to make the color of the skin black, the information it was transmitted from parents to the offspring. So this trait is heritable. It means it can be transmitted from one generation to the another, right? So now, if so, then these things, I expect these things might have been already discussed in the previous discussion as well. But I'll just refresh and go back go to the questions of what causes the variations. I wanted to discuss this as well. So for a natural selection to act, what are the essential requirements? So variation is very essential. Here, the variation in terms of skin color is important. Now, the second point, which is very important, is the heritability. Heritability means the transferring of the character from the one generation to the another, and differences in the fitness. So when I say fitness, what it means is the ability of an individual to survive up to the reproductive age and to reproduce. The individuals which has a higher probability of surviving and reproducing, they will become more fitter than the other one. So here it's not always that the bigger are fittest, or it's the heaviest are the fittest. It's not like that. Okay. But any individuals in a population who has a better ability to survive relative to others in the population and reproduce, they will have a higher fitness. So there should be a difference in respect to fitness for a natural selection to act. So this is the mechanism which Darwin came up with to explain the theory of evolution. Now the question again is like, okay, it's very apparent for us that, by this time, that there is a variation with respect to traits in population, but how these variations are coming up, and how these traits, how this character is being transmitted from one parents to the offspring. So this question was very important that time as well. Now Darwin came up with the idea of this pangenesis to explain how the characters might have been transmitted from the parents to the offspring. So basically, what he's trying to tell in this idea is so during the gamete formation, so there will be a thing called gamete. He called it gametes. It's basically a particle. It's basically some biological material that will be collected from different body parts. It will get collected in the gonads, and it will be transmitted to the offspring. So that was the idea of pangenesis. So the overall process is called pangenesis, and this is how Darwin tried to explain how characters might have been transferred from parents to offspring. Now, this idea is no longer valid now. Why? We'll discuss. Now, with respect to heredity, okay, so this we'll discuss later also. Now, so in case of natural selection, if we check, then there was not only differential reproductive and differential survival ability, but the character need to be inherited to the next generation. So during those time when Darwin was proposing his idea, during those time there was a strong opinion about the blending inheritance, how character will transmit from how Mendel tried to explain versus how this blending inheritance idea explain the inheritance of the trait. It's completely different. So through this example, I like to explain what the blending inheritance means in a simple term. So let us assume that we have a plant. Let us assume that we have a plant in which there are few individuals which height is very short height, and the few individuals having a tall height. Now, while running the experiment, we can actually control the pollination, control the fertilization. So what we can do is we can collect the gametes from the tall plants and fuse it in the short plants, or vice versa. So if we do so, then again, say this is a plant. This plant will have a flower. What we'll do is we'll collect the gametes from the flower, we'll fuse it, and then we'll wait for the flower to fruit. And then when that fruit when it is fruiting, then again we'll collect those fruits, we'll sow that, and we'll see how tall or how short the plant is there in the afternoon generation. So here this is the plant that came up from the seeds which was sown. They were collected from this pollination, and then they grew this, and what they could see is that basically what this blending inheritance tell is that this character, this height of the plant, it mixed, it just blend. Okay, so for example, if I'm making this confusing, then let's assume that there are two plant with two different colored flower, one red and one white, and if we mix the gametes between these two plants with two different color, then in the next generation we expect plant to have a mixed colored flowers. So even within when they self-fertilize, what they see is that all individuals were having a medium height. So here, this tall character and the short character, it got lost. It just lost in the lost over the subsequent generation. So that was the idea of blending inheritance, and because of this, the variations that we see initially in the population, it is now no longer existing there. It just blend. So because of this, so the idea of heredity was like this during those times. But in the similar time itself, there was a scientist named Mendel. He was doing experiment with the garden pea plant, and he could discover that there is a entity, there is a particular character that is being transmitted from the parents to the offspring, and that character is transmitting as a separate entity, as a factor, and it's not blending or it's not mixing. So that was the idea that Mendel came up with when he was running the experiment. So I hope that some of us are aware of the various traits which Mendel took considered for running his experiment. So he considered numerous traits. When I say trait, it's a character. To make it more simple, when I say character, it could be anything, any character that is of our interest. For example, in this case, maybe the shape of the seed is a character or a trait. Color of the seed could be a trait. So likewise, he took many traits, many characters, and what he did was he did an extensive experiment. He did the pollinations. He did this cross between individuals with round colored seed and individuals with wrinkle color seed and like all those things. And what you could find is that over a time the variations are maintained over time. So what blending inheritance was discussing, or it was discussing, was that over a time, even if the parents they have a different phenotypes over a time, because the traits are getting mixed, the traits are getting blended. So because of blending, the variations will be lost over a time. But Mendel experiment could show that it's not that way. So variations are maintained over time. So it's a discrete. Characters are not blending. So this is the idea which came up with the experiment by Mendel. But but we know that for a long time the idea which was developed by Mendel, it did not got a lot of attention from the scientific community. So that we know, and only during the 1990s, when scientists started performing experiments with various plants, they could get the pattern similar to what Mendel got in his experiment. So then again, his idea was his idea got popularized, and it led to various subsequent development in genetics and understanding of how variations comes into being. Now, I thought I should again take the help of this diagram to discuss briefly about the differences between the blending model and the Mendel inheritance model. So this is again here one individual, one parent, let's say, having a tall height, and one individual is having a short height. So when there is a cross between these two individuals, in the offspring, in the F1 generation, what we can see is that all individuals are having a medium size height. Even when we self-fertilize them, what we are getting is we are getting all medium size height. So here, what we can infer from this is that the tall character and the short character that we see in the parental generation is now lost. They just blend, the character blend. Now, in case of Mendel experiment, the pattern that Mendel saw, so when he crossed these individuals with the tall height and individuals with short height, what he got is, F1 generation, he got all individuals with tall plants. And there was no short plant in the F1 generation. But when he self-fertilized means when he collected the pollen from the same flower and and fertilized the gametes, then in the F2 generation, what he got is he got 3 tall plants and 1 tall plant. The ratio that he got is 3:1, tall, 3 tall, and 1 short. So here the major difference between this blending and the Mendel actual results are, so in this case, this trait, tall character, is represented in both F1 and F2. That's fine. But importantly, this F2 in F2 generation, what we can see is that this dwarf character or the short character also is getting expressed. So here the character, it's not that tall character and short character they are mixing and making the medium character. It's not happening like that. Tall character is there, dwarf character is there. So in this case, what we can also understand from this is that even when all individuals in this case, all individuals were tall, yet there was a character. There was the information to make this short plant. It was there in this tall plant. Simply, it didn't express. It didn't express means you have a genes. I thought I'll come to the gene later. But anyway, you have a gene or the information, but it is not getting expressed. It's there, but it's not mixing up. So that's the basic difference between the blending hypothesis and the Mendel inheritance pattern. Yeah, as I discussed, for a long time, Mendel work was not it did not get it was not popularized, but with the independent work of Hugo de Varese, Calcorens, and Tisselmark, the idea of inheritance, which was proposed by Mendel, was rediscovered, and then it led to subsequent multiple different experiments and different findings. they did work similarly. The other scientists also tried to replicate the kind of experiment which Mendel did in the different taxa, different model system. When I say different model system, means like some of them try to work on plants, some of them try to work on animals, and see if the pattern that was shown by the Mendel, it still whole exists for still exists in this different model system or this different species. So in all of these cases, what they are finding is that the variations that was there in the parental generation, it's they are maintained over time, right? So it's very important observations. Now, during this after the work after the work of Mendel, August Weismann he came up with the idea of germline theory. Okay, so now I like to go back to this idea of pangenesis once again, just to make this thing clear. So pangenesis, according to this pangenesis theory, what was the explanation was this. So each of these like the information that is passed from parents to offspring, this information is being collected from different body parts, and it get collected in the gonads and it is transferred to the next generation. So, for example, the color of the eye, how the offspring, like what will be the color of the offspring eye, to decide that to have the information, the something called granules will be collected from the eyes. I mean, what Pangenesis is saying, that there is some biological process so that the information is being collected from the eyes of the individuals and will get collected in the gonads. Similarly, what will be the hair type? The information for hair type will be collected somewhere near the head, and it will again get accumulated in the gonads. So such was the idea of pangenesis. But here, this August Weismann, what he is trying to explain is this. So there are two different types of cells in the body. One he is calling a somatic cells. Another one he is saying a germ plasm or gametic cells. So somatic cells, in a very simplistic term, we can understand it this way. So, for example, the skin, the cells that make our skin, or let's say the muscles, or the head, all these things. So these are the somatic cells, and the cells that makes the gametes, they are the gametic cells or the germ plasm. So what he's saying is that the information from parents to offspring, it is being transmitted through the germ plasm, and not the information is not being collected from the other body parts. For example, not from the other somatic cells. So this was a very important understanding that came up after the Mendel work, and so this also refuted some of the ideas that was proposed or that were in discussions about the Lamarckism. That is particularly the inheritance of the acquired character. So when I say inheritance of acquired character, to explain this again in a brief, I like to take one example. Let's say there is a mice, and in the lifespan, when the mice was growing up, somehow accidentally its tail got chopped off. The tail was cut down because of some accident. Now, when this individual mice reproduce, whether the offspring will have a half tail, or let's say they will have a complete tail, or so. If this inheritance of acquired character exists, or if this inheritance of acquired character is there, then what we expect is the offspring that is produced from the individuals which had a half tail will also have a half tail, or something like that. So this inheritance of acquired character was there in the discussion, but after the discovery of this August Weismann germ plasm theory, after the understanding of this germ plasm theory, this idea of inheritance of acquired character was getting sidelined. Was actually people had an answer to refute. People had rational argument to refute this idea of Lamarckism. Yeah, and August Weismann also strongly supported the idea that the natural selection is actually a very creative agent that leads to evolution, and and he didn't give any importance. And like when I say he didn't give any importance, it doesn't mean that he simply didn't give importance, but based on the rationale that he was developing, based on the experiment that he was conducting, so he didn't give much importance to the inheritance of acquired character. Now, so today's discussion is more on like Neo-Darwinism. So I thought like at least I should introduce the scientist who invented this term Neo-Darwinism. So it was George Romans, and he invented the term Neo-Darwinism. And basically, this is to explain the Darwinism that was advanced by August Weismann. And later on, again, like numerous other scientists came and they improved it further. But Neo-Darwinism was not very popular initially, but once the Fisher and J.B.S. Haldane and other scientists came, and when they came up with their different experiments and different results, then it became more popular in the 1930s. So the point is, the variation is important. For a natural selection to act, variation is important. Now we'll quickly look into what causes these variations. Okay. Now. One of the scientists who rediscovered the Mendel work, Hugo de Vries, he was conducting an experiment on evening prime rows, and while conducting the experiment, he got very unexpected results. Basically, he was conducting an experiment with a purebred, basically like individuals which can produce the same type of individuals in the subsequent generation. For example, if I grow a tall plant, all the subsequent generation will be tall. So we call it a true breed. So he was also running an experiment with a true breed line of evening prime rows, but sometimes he got something very unusual flower type. For example, let's say he was doing an experiment with flower with a plant with flower color X, and in few generations he was getting plant with flower color Y. Let's say. So he wanted to understand why this unusual pattern coming up in this experiment, in this kind of experiment that he was doing, and he introduced this idea of mutations. It could be that there is some changes that's happening that might be leading to these different phenotypes, even in the true breeding lines. Now, so at this moment, when he was doing this experiment, and at this contemporary time, so the idea that the unit of inheritance is it's physical. The physical unit of inheritance is more or less called gene. That was coming up, and genes are particulate. It means it's not blending, which I tried to discuss previously. It's not blending. That was coming up. So now at this moment, after the slides, I like to focus on like what causes the variations. Okay, so there are basically two sources. Again, when I say two sources, it's again in a simplified form. I'm saying two sources. One is genetic variations, another one is non-genetic sources of variations that we see that is leading to a variation that we see in the populations. So first we look into genetic variations, and there are multiple sources that lead to genetic variations in the population. Again, so here when I say genetics, it means I think I should have included one slide anyway. So just to make it so there is a gene. It contains the information, and based on the information, the character is expressed. Definitely, there is an effect of environment, but gene definitely has some information. Based on that, the character is expressed. For example, in case of the example that we took for the color of the mice, so there are some information that is coded in the gene, and based on the information, some individuals are becoming black, and some individuals are becoming green or tan. So that's a gene. So gene is basically a unit of heredity. So that gene might undergo mutations, and that mutations might lead to variations in the population. Another one is the gene flow, and another one is sexual reproduction. So that these two I'll also try to discuss. So so mutations are generally considered to be a raw material of genetic variations, and there are again two types of mutations that we can see in our in living system. One is somatic mutation, and another one is germline mutation. The mutation that's happening in somatic mutations might not have very significant effect in changing the composition of a phenotype over a generation, because it's not transmitted. to the subsequent generation. But the mutation that's happening in the germline, basically the mutation that's happening in the gametes, the cells that makes up the gamete, those mutations are inherited to the next generations, and that lead to the change in the composition of the actually that lead to the change in the composition of the alleles, and that eventually lead to the change in the composition of the phenotypes. So yeah, just to give a brief, just to give an idea of what it means by germline and somatic cells. So just to give example, the skin, it's a somatic cell. The cells that make up the skin, or let's say the muscles, in the somatic cell. The cells that make up the gametes, they are the that's a sorry. That's a germ cells. Yeah. So what kind of mutation exists? So again, there are broadly there are two types of mutation that exist. One is gene level mutation, another one is chromosomal mutations. Now, what is chromosome? Chromosome here means basically, like, we have a cell. We know that our body, we are made up of different cells. We have millions of cells. Each cell has a nucleus, and inside nucleus there is a thread-like structure. That thread-like structure is made up of the DNA, and it also has a protein. So the thread-like structure, which that's there inside the nucleus, is what we call chromosome. And it's always not necessary to be inside the nucleus, but for this discussion, yeah. So I'm trying to explain that way. Now, what kind of mutation exists? So gene level mutation, I'll try to explain gene level mutation here. So the genes, when I say the gene is made up of different nucleotides. When I say nucleotides, it's basically a different compounds, different molecules. So these different molecules are represented by different letters in the scientific world. For example, some compounds are known to be like represented by letter G, some are C, and all those things. Basically, these are this is the chemical name, this is the compound name, adenine, thymine, guanine, cytosine, and all those things, and they are represented by these different letters. So it could be that this letter they might be mutated sometimes. So the types of mutation can be of different types. For example, sometimes there could be a substitution mutations. When we say substitution mutation, basically if there is a in the sequence of a DNA, and if it is substituted by C, then C or any other nitrogenous base, then that will be a substitution mutation. Similarly, some of the letter might be deleted from the sequence, then it's called deletion. And sometimes new code could be added, new base could be added. That is called insertion. And there is inversion, basically when there's a flip in the sequence, that's called inversion. So this is a gene level mutation that's happening at the DNA level. So why this is important is because, again, I told you the information for what type of character being reflected or what phenotype will be expressed is there in the gene. So if there is a mutation, if there is change in that information, then the phenotype, the character that we see in the individuals might also vary. Again, I'll like to add here that the environment too play a role. It's not always the gene, but environment too play a role. But to make it a simple, I'm focusing more on the genes. Now, there could be a similar, not similar, but there could be another mutation called chromosomal mutation. Basically, what it means is, let's say, for example, in case of human, we have 23 pair of chromosome. That is total 46 chromosome, and sometimes there will be a change in the number of chromosomes which individuals have. For example, let's say 47 number of chromosomes, or let's say if individuals have only 45 number of chromosomes, then that's called chromosomal mutations. But this is not the only one. There are other types of mutations. For example, deletion, duplication, all those things can be there. All this called structural mutation is also there. Similarly, there is another kind of mutation which we call numerical, which I just discussed now. For example, let's say Drosophila have, let's say, X number of chromosomes normally, but because of the mutation now they have Y number of chromosomes. Then that's also a chromosomal mutation. So there's a change in the number of chromosomes. So all these things might result in the different phenotype expression. Now, what causes this mutation? Okay, so it seems that there are different level of mutations. Some are somatic, some are germline. The level of mutation could be within gene or within chromosomal level, but what causes the mutations? So there could be multiple factors which lead to this mutation. One is spontaneous. When we say spontaneous, so there are different biological processes that's happening in the cell level, in the nucleus level as well. So what we call, like there is a DNA which has information. Now it should be copied in it should be copied in the form of RNA, and then from the RNA, the information will be copied in the form of protein, and that protein will bundle up and make up the structure, right? So in the process of this copying from DNA to RNA and then to protein and making the phenotype, there will be an error. That's one example of spontaneous mutations. The DNA that we have in our cell, the cell in itself need to divide. Okay, so when it's dividing, there also the DNA need to replicate. Replicate is basically DNA need to make two copies, and in the process of making this copy as well, there is a chance of some random mutations coming up, the spontaneous mutation coming up. That could be another reason, another ways. So similarly, the mutagens, there could be some chemicals, there could be some factors that will lead to the mutations. So they are called mutagens. That may also lead to the mutations. So so I think at this point, definitely I know that in this short time, it's very some of the term that I use may be technical, but what I'm trying to tell you is, so there are different ways by which there can be changes or there can be alteration in the genes, and that alteration in the genes might be reflected in the phenotype, and might be reflected in the character that we see. For example, the color of the coat of the mice, or maybe the color of the seeds, or the height of the plants, or all those things. Now, so till now, my idea of this talk was to discuss these different types that will lead to a change in the genetics, and also to discuss the various processes which will lead to which will change the composition of the phenotype in the population. So definitely, we already discussed this idea of natural selection. Again, I don't want to discuss it because we don't have much time. Now I'll go to the another mechanism, another process by which the composition of the phenotype in a population might change. Okay, so and that's what we call gene flow. So the name itself is very explanatory here. So let's assume that here with black or brown beetles, and there is another population B, with the green colored beetles. Now, just to give a clarity, these two populations belong to the same species. It means if I put this brown and green beetles together, then they will be able to produce the viable offspring, and that offspring will live up to the adult stage and reproduce. So they are of the same species. Now, so let's assume that this is population A and population B, and here if you see, then there is no green color individuals, right? But what if the individuals from this population move to this? What if they migrate to this population? Now, here in this population, the composition of the phenotype, it will change. So the ratio of the green versus brown will change here. And similarly, if few individuals from here moves there, then again the composition of the phenotype changes. So gene flow is also one mechanism by which the phenotype in the population changes. Now, there is another concept called genetic drift. So to understand this, let us again take this example of the beetles. Okay, so in this case, we can see that there are two different color beetles. Again, they are of the same species, and they are of the same population. It means they are located in the same geography at particular time, right? So here, what we can see is that the phenotypic composition here is 1:1, 3 green, 3 brown. Now, let us try to understand what genetic drift is. So let's assume that they are there in our garden, and when we are gardening our garden, when we are weeding out the weeds, we accidentally step onto like two beetles here, and that two beetles was accidentally happened to be green color. Now, what will happen in the population? In the population, the ratio of green versus brown, it will change, right? So this is called genetic drift. So here, the compositions of the phenotype in a population is changing, and the factor that is causing is a random factor. It's here the accident. Okay, and this way also the compositions of the phenotype in population changes over time. Now, the sexual reproduction is definitely one way that will lead to variations in the composition of the phenotypes, and there are different processes that involves in the sexual reproductions, right? For example, there is a thing called meiosis. Meiosis is basically a type of cell division in which there are multiple steps involved that include crossing over and all those processes. And during the process, uh, the way the genes are getting segregated, the way genes are getting segregated, and how chromosomes are getting aligned and getting separated. Based on that, the phenotype in the subsequent generation might the composition of the phenotype in the subsequent generation might change. Yeah, but see, okay, I also wanted to discuss this part again, since I work in butterflies, so that's why I wanted to put one slide with the butterfly. So there's a variations in the variation that we see in the populations. It could be because of genetic variations, but there are non-genetic source of variations as well. For example, let's say I'll take the example of this butterfly. So we all know that butterfly have a different life stages from egg to caterpillar to pupae to adults, and here in this that this photograph here in the left, this is the pupae of the same species. Okay, and all this color that you see from green to dark brown, all these pupae are coming from the same parents, or maybe like they have a similar genetics, but they have a different color pupae. And this variation in the pupa color is because of the environment that they're exposed to during this process of pupation, right? So if they are in the black background here, it's likely that they'll become brownish. If they are against a green background, then it's likely that they'll be green. Similarly, this is another example where we can see that the variation. We can see that there is a variations in the color of the skin, and that is induced by environment. They have a similar genotype, but based on what environment they are experiencing during the development, they can either become a green or a brown. So when we see the variations in the population, it's not always necessary that variation is caused by genotypes. It could be environmentally induced as well. For example, let's assume that we took two seeds from the same parent, and we grow them one with all the all the nutrient available. We provide them all the nutrients, water them, and all those things we do. And for the another one, we are not watering them, we are not giving them nutrient. Then in that case, even though both of them have a similar genetics, but they will not have a similar phenotype because the environment is having a role here, right? So it's not always necessary that the variation that we see in the population is caused by genetics. So and like here, yeah, definitely, this the ability to have a different phenotype when they exposed to different environment, that itself is caused by genotype. That there should be the genotype must have the ability to produce different phenotype, but it's not entirely. But like the environment has a strong effect here. Now, this after the development of after the development of the idea of the genetic mutations and variations and all those development, many scientists came, particularly in the 1930s. For example, Thomas and Morgan, we did extensive work on Drosophila, and he could find he could come up with different discoveries. Fisher and Dobzhansky, Haldane, and Swellright, all these people came up with a different experiment. They did different experiment. They come with different ideas, and it led to the development that today what we know is a modern synthesis is because of the contributions to many scientists. Some of the names are represented here.